Hello everyone, welcome to week number 5 in Bio 120. We are past the halfway point now. I want to say happy Thanksgiving to everyone, and I hope that you get to celebrate it in some way by gathering with your family and friends, by having a special day, quiet evening, whatever it is that you do. I hope that you get a chance to celebrate it this week and give thanks for everything that you have and everything that you are. All right, with that being said, let's see what we've got going on. Last week was all about metabolism and photosynthesis, and in metabolism is contained cellular respiration, which is the way that we break down food in order to make ATP molecules, the energy molecule that runs all of our biochemistry. So we covered cellular respiration, we covered photosynthesis, which is a way that autotrophs make their own glucose by using light energy and carbon dioxide and also water to produce glucose molecules. And you talked about whether cellular respiration and photosynthesis were opposite reactions, complementary reactions, not related. And of course they're complementary reactions, and you know this because in plants and other autotrophs, they do photosynthesis and they also do cellular respiration. So they have to be complementary. All right, for this week is starting our genetics section and also the cell cycle, so the life cycle of a cell. So we're getting into that in just a minute, and I'll talk about that picture over there in just a second. Skim through chapters 10, 14, and 15. Cell reproduction, how DNA replicates, makes a copy of itself, and chapter 15 is all about the job of DNA. And that's a really important concept to get, because why is DNA genetic information? Because the code is there to make all the enzymes that you need as an organism to run all your biochemistry, to make the molecules that you need, to break down the food substances that you take in as food, to do everything that you need to do. So that's the job of DNA, to code for all these different proteins that are enzymes that run your biochemistry as an organism, the species that you are. So those are very important chapters. Just skim through them though, because remember, by using this online textbook, you can search anything that you need to, which is an awesome feature of this free online textbook. Read the Module 5 overview. It's pretty good. Watch the videos, especially the one on the cell cycle and cancer. So watch that one. But do watch the videos. They're short and they get to the point. And then, so you've got some assignments due. Your discussion board postings are, your initial post is you're discussing cancer with a friend of yours and you're having a debate about whether cancer is a problem with cell division, because there's something abnormal, right, if you develop cancer cells, or is it a genetic disease? So are there genes that lead to cancer production? To start off with, what is cancer? Cancer are your cells that are dividing abnormally out of control and are taking up valuable resources and energy from the rest of your body tissues. The most important thing that cancer cells do is that they metastasize, and that means they move to other parts of your body, set up in that location, take your resources, nutrition, energy from that location. And so that's the dangerous part about cancer. So here's your discussion with this person. And when we look at cell division, it's important to understand the cell cycle. So take a look at my little circle over here that I drew of the cell cycle. So the cell cycle is just the life cycle of a cell, right? Yeah. So it goes through different stages, just like we do as human beings, right? It goes through different stages. It stops sometimes. It checks itself sometimes, and that's what those slashes are, to make sure that everything is going right. So we start at 12 noon, okay? That straight up and down line is the start of when the new cell has just been produced through the division of other cells, right? So that's when we start. And we move into the G1 phase of the cell cycle. G1 is a time of general growth. So enzymes are being produced that are needed. New organelles are being produced so that you have enough mitochondria, you have enough of the Golgi apparatus, the endoplasmic reticulum, the ribosomes, all the other organelles that you need. General growth happens in G1. I'll get back to G0 in just a second, okay? So general growth happens in G1, and then you get to a checkpoint. So checkpoints are just that. They're where the cell checks itself to make sure everything is good before it moves into the next phase. So that's important, especially with our cancer discussion here. The next phase is the S phase, and this is where the DNA is synthesized, and that means that the DNA replicates, makes a copy of itself. That's very important if the cell is going to divide. It has to have 2 pristine copies of the DNA in order for this cell to divide into 2. So that's the S phase. Then it moves into the G2 phase. In G2, all the cellular machinery that's needed for cell reproduction, like the spindle fibers and the centrioles and lots of enzymes that you have to have and all that sort of thing. That's when those are produced. Notice that there's also a checkpoint between the S phase and the G2 phase, another stopping place where the cell checks itself, makes sure everything is okay before it moves on. And then you get up to where it says M and C. So M is mitosis. It can also mean meiosis if these are sex cells, but this is where the nuclear division happens. So all the DNA gets split up, right? That's M. And C is cytokinesis. That means the rest of the cell splits apart into 2 parts. And then you get back to 12 noon, and now you have 2 daughter cells, and they each go into the G1 phase. Okay, one other thing to understand is that the job of the cell is not to make copies of itself. The job of the cell is its job, right? Well, you haven't described anything in there. Everything's been about cell division. Yeah, that's where G0 comes in. So enough growth has to happen in the cell, enough new enzymes have to be produced, you have to have enough organelles, all that sort of thing, for the cell to do its particular job. Like a heart muscle cell, that cell pumps, right? It contracts. Let's think of another cell. A brain cell stores memory. Let's see. A small intestinal cell absorbs nutrients. So there are jobs that each of your cells have, and each of any organism's cells have. And so in G1, there is this stopping place called G0, and that's where the cell does its work. That is a very important part of the cell cycle, G0. It's where the cell works. Okay, so you got all these checkpoints. In cancer cells, all these checkpoints are completely ignored. So all kinds of wacky things can happen. The cell doesn't have to have everything in place for it to actually split and make 2 new cells. And that's what happens with cancer cells. They get all kind of cattywampus and crazy, and too many chromosomes and not enough chromosomes, and the cell's too big, and all kinds of strange things happen with cancer cells. And that's because cancer cells ignore those checkpoints. So that's a problem with cancer and cell division. Then the genetic disease question and the genes. I know that you have heard of cancer genes before. They're tumor suppressor genes, they're proto-oncogenes. Read about those in your textbook. There are genes like the BRCA1 and BRCA2 genes that can lead to breast cancer and or ovarian cancer in women, also in men. But you've heard of those genes before, so you know that there are genes that can cause cancer. And there are many, many more that we know now. So here's your discussion with your friend, and you're going to talk about this. Is cancer a problem with cell division? Is it a problem with genes? And you're going to figure out why these two components are really important for cancer to happen. And like I said in another week, if the question ever says, is it this or that or both? Yeah, that was last week. It's always both. Otherwise, no one would ask you that question. So just know that. And how would lifestyle changes lower your cancer risk? To 0? Never. That isn't going to happen. You live out there in the world. You have predispositions to cancer. So, yeah, you can never lower your cancer risk to 0, but you can lower it. So, what are lifestyle changes that you can make happen to lower your cancer risk? And then finally, in the response post, you're going to talk about environmental carcinogens. So, a carcinogen is anything that causes cancer, like benzene, like cigarette smoke, like alcohol, like oh my goodness, there's so many drugs. And I'll give you an example. One of my aunts had horrific pregnancies way back in the 50s and 60s, and she got tremendously nauseous. So her doctor gave her a drug called DES, and it did cure the nausea that she was having. Now we know that exposure to DES in the womb makes any offspring susceptible to cancers, and especially female fetuses, and then the female offspring. This can lead to breast cancer and ovarian cancer. That's an environmental carcinogen. All 3 of her daughters were diagnosed with breast cancer. My first cousins, all 3 developed breast cancer because of her exposure to DES. That's an environmental carcinogen. And how can we remove these environmental carcinogens from the environment so that we're not exposed to them? Yeah, that's really important. Okay, so those are your discussion board postings. Let me move my face over. I'm always into letters. Okay. Then you have milestone 3, and here we have like the busiest week. It's a tremendously busy week for me. I'm cooking for 15 to 20 people. So yeah, it's a crazy week this week for me, and it probably is for you. But you have the most important milestone of the whole final project this week, because it's all about the biology. Yeah, and that's what you're supposed to be getting from this course. So anyway, I'm going to help you with these. I'm not giving you the specifics for each one of these categories and topics, but I want to mention a couple of things so that you get the right idea. So in milestone 3, you're going to cover the methods of conservation, and this means prevention. How can you prevent biodiversity loss? How can you prevent air pollution? How can you prevent GMOs from causing harm? How can you prevent climate change? Okay, prevention. That's A. Education. Why is education so important about this topic? Do you remember being educated about any environmental issues when you were in school? I know I was. Oh my goodness, I was a crazy fanatic. I was. This is when we were just first understanding about carcinogens, really. And one of my science classes, the teacher gave us a list of carcinogens in foods like nitrites and nitrates in processed meats. Now, I grew up in a Southern household. We love bologna, hot dogs, all that. Yeah. So what did I do as a kid? I put that on my mother's refrigerator. She left it up there. So anyway, education is really important. Then C, solutions. What are the fixes? Now that we already have this problem, how can we fix this? Totally different from prevention, right? How do we fix it? D, ethical considerations, including laws, regulations, any kind of conservation groups that are in this mix having to do with this topic. What are the ethical considerations having to do with your topic? Because there are ethical issues. there's a reason that we have this problem, and there's a reason that we haven't fixed it. So what are those reasons? Positive and negative aspects of the issue. Is there anything positive that comes from burning fossil fuels? Not in the long run. So we'll see. What about the negative aspects of the issue? And then personal impact. What can you do as one person to help fix this problem? Right? What can you do? Don't be a litter bug. Ooh, I remember that from growing up. Don't be a litter bug. Don't dump your trash out on the road, right? I think we need to go back to that one, because I sure do see a lot of litter in this country. What's the personal impact of this particular topic? How can we change things by just the actions that we can do? Include citations and references. This time, this is not an outline. These are PowerPoint slides. 6 to 10 slides. Do not just write out the words on there. You can always put the written out information in the speaker's notes, which is at the bottom of the slide, right? If you don't use PowerPoint, you can make a PDF presentation, you can use Canva, you can use Google Slides. There's all kinds of applications out there and ways for you to make slides. So PowerPoint slides this time, and if you don't, you get points counted off, so just know that. And label all your sections. Methods of conservation, that should be on the top of the slide. Education and why it's so important. Next slide. You get it. Label all your sections. I shouldn't have to guess what you're talking about. And boy, I've seen some where I just had to guess, and sometimes I didn't guess very well because the topics weren't covered completely. So make sure you label everything. Slides, cover all the information. And if you have any questions, if you're not sure, if you get stuck, like if you're really not understanding the difference between prevention and solutions, I will be glad to discuss that with you through email. Just let me know. Or, we can set up a face-to-face meeting through whatever I have, WebEx. Zoom call, something. We can set up some kind of meeting. All right, and the last thing, you do not have to stay for this part if you don't want to, but since this week is all about DNA, and I love DNA fingerprinting, that's the old terminology. It's now called DNA profiling, typing. Yeah, there's all kinds of names for it now. But when I went to graduate school, this particular technique was new, and I did my dissertation on aspects of the DNA fingerprinting technique, like the one that you see over there on the right side of the slide. So I'm just going to give you a little bit of history about this particular aspect of forensic science, and it has many other applications today too, but it is used a lot in forensic science. So I just want to cover a little bit of the history. In 1985, Sir Alec Jeffreys, before he was Sir Alec Jeffreys, he was just Dr. Alec Jeffreys, published a paper on the use of DNA analysis in order to tell the difference between individuals. And he used very closed communities of people and the family structures to see if he could tell the difference between people by doing this particular technique. He chopped up the DNA. He labeled the DNA in areas where there was variability, right? Wouldn't that make sense? You got to find the places where there are differences between people, because there are lots of DNA that we have that's exactly the same from one person to another. And then he made sort of a barcode, and that's what you see on the right side there. That's an example of what he would have seen. So it does look like a barcode, right? His wife, when he explained it to his wife, she said, "Oh, this is kind of like fingerprinting." So he coined the term DNA fingerprinting. Well, he was in England, and the police were very, very interested in this technique because they had 3 murders on their hands of teenage girls, and they had no idea who had committed these 3 crimes. And so they went to Alec Jeffries and they said, could you possibly use this technique if we brought you crime scene samples and evidence, and then we collected blood from men in the adjoining areas? And he said, yes, it should work. So he started testing. Now, in England, they don't have the same civil rights that we do. The police can just go out and say, every man in this village, come forward, we need to draw your blood, and that's what they did. They drew blood from about 5,500 men, and he started running the samples against the evidence that he had, and, time after time after time, no, he wasn't coming up with anybody. And then something happened. One of the men in one of the villages convinced his friend Ian Kelly to give blood a second time and say that he was Colin Pitchfork. Honest to goodness, that is the man's name. He said, I've been in a little bit of trouble. I don't want them to look at my blood. Ian, please, would you give blood for me? And Ian Kelly said, sure, I'll give it for you. So he went a second time and posed as Colin Pitchfork. Well, later that day at the pub, old Ian had a few too many pints, and he started telling everybody in the pub, what he had done. Well, one of the women got pretty suspicious. She said, why would Colin Pitchfork do that? And so she went to the police and told the police, and they went to Colin Pitchfork's home. He was a very respectable family man, had a full-time job, never been in trouble with the law, didn't find anything in his house, but they brought him in, they took his blood sample, and sure enough, his DNA matched that of the evidence. He is now in prison still, since that time, in prison with a life sentence in England, because he did indeed murder those 3 young girls. In 1988, the U.S. got wind of all this. They had been reading about this phenomenal technique, and the FBI started using this technique. Now, this was called the RFLP technique. Restriction Fragment Length Polymorphism. You don't need to know what that means, but it means it makes a barcode-looking radiograph that looks like this on the right side. What I've given you here is a mother's profile. You look in the columns. A mother's profile is on the left, M, mother. Child, C, is in the middle, next to the mother. F1 is the first possible father. F2 is the second possible father. And so the way that you read this is you go down the column, the child's DNA pattern, right? You go down the column. You look at their first band. That band has to be either in the mother's lane or the father's lane, or both. It can be both. Okay, so you go down. You look at the second one. Okay. There's no band in the mother's lane, so it has to be in the father's lane. So F1, no, he doesn't have that band. F2 does. Okay, keep going down, keep going down. Okay, there's one in the mother's. Okay, keep going down. Okay, there's a nice thick one. Yep, over there in F2. It looks like F2 is the father of this child. So that's a paternity test and how you used to read that. Then along came something called the polymerase chain reaction, which magnifies and replicates certain segments of the DNA that's variable. And so now you didn't need as much good intact DNA. You could use very degraded DNA. We're still using this type of technique today, but it's so much more sophisticated than in the 1980s and 90s. But we still are using this particular technique to magnify and replicate DNA in certain locations. This is what we use now. So we can use much tinier amounts of DNA, much more degraded. And so that's what we use now. And we don't get a pattern that looks like that on the right. And finally, in the last few years, new things have been happening with forensic science and DNA. Genealogy websites have been used to analyze DNA to help catch criminals. This is called forensic genetic genealogy. And so it's a new type of forensic science, which is very exciting. I find it extremely exciting. The first case that was ever publicized about this was for the Golden State Killer, Joseph DeAngelo, who had been free for about 40 years. He had assaulted and killed quite a few people all up and down California, and had never been caught and really never even been suspected. And he was convicted based on forensic genetic genealogy. So that's very exciting. So that's just a little bit about forensic science and how DNA is used in forensic science. And it's getting more and more specialized, and we can do more and more things with it as we learn more about DNA and all these different techniques. And if you would like to discuss this with me, feel free to email me. I'd love to get into a discussion with you because, of course, this is right up my alley. All right, with that being said, I hope that you have a wonderful Thanksgiving week and that you don't get too stressed out. And if you get stuck, like you're out of town, you can't get to your computer, just let me know. You know, I'm flexible with the time, especially this week, because it will be a crazy week for me and for you. Just let me know. With that being said, I hope that you have a wonderful week. Let me know if you need anything. m.sigmon@snhu.edu.